The boundaries of life are blurring. Jacob Hanna's synthetic embryo models and the three major controversies. Opening, posing the philosophical life question. Imagine that the origin of life, the miracle that begins with a single fertilized egg, has now been completely decoded by scientists. They didn't use sperm and egg. Relying only on the most basic cells, they modeled structures with a spine, digestive tract, and even a beating heart. In a top laboratory in Israel, Professor Jacob Hanna is attempting to answer the most monumental question humanity has faced for millennia. Can life be artificially created and controlled? His team has created synthetic embryo models with incredibly high levels of organization. How similar are they to true early embryos? What scientific limits have they challenged? And how will they completely change our understanding of reproduction, disease, and death? 2. Act 1. Hanna's breakthrough technology. Limitations of traditional methods. In the past, studying early embryonic development was a huge challenge. We relied on animal models or donated embryos, which have enormous ethical restrictions. These methods were often a black box, allowing us only to see the outcome, but not to observe the process. The core of Hanna's technique. This is the real game changer. Professor Hanna completely bypasses the fertilization process. His raw materials are pluripotent stem cells, known as iPSCs, commonly referred to as master cells, capable of becoming any cell. Through an ingenious combination of chemical signals and mechanical environments, they successfully awaken the cell's self-organizing instinct, allowing them to begin self-designing and self-constructing like a true embryo. Result demonstration and detail shock. The result of this self-construction is shocking. We are not just looking at cell clusters, but structures differentiated into clear tissue layers. A neural tube, which could form the brain and spinal cord. A yolk sac, responsible for early nutrition. And even, we can clearly capture a heart-like structure beating rhythmically within the model. These complex structures have been built from scratch, without the involvement of the natural life process. 3. Act 2. The immense potential and application of the research understanding miscarriage and defects. The first immense potential of this model, understanding the causes of human developmental failure. Why do some people experience recurrent miscarriages? Why do congenital defects occur? Previously, we couldn't study these in the early stages. Now, these synthetic embryo models offer a clear, repeatable window of observation. The revolution in drug screening. Drug screening is about to be revolutionized. We can use these models to test the potential risk of new drugs on fetal development, avoiding teratogenicity. This is closer to human response than expensive animal experiments and is also more ethical. Organ regeneration and future medicine. In the long run, Hanna's technology is a key step in regenerative medicine. If we can understand how to build an embryo model from basic cells, then in the future, we may be able to precisely repair or regenerate complex tissues and even provide a blueprint for rebuilding organs for humans. Act 3. Ethical boundaries and scientific accountability. The core ethical debate and controversial viewpoints. Scientific progress has never generated such intense moral pressure. When discussing these synthetic models, controversial questions immediately surface. Skeptics argue that this asexual reproduction model dangerously approaches the creation of life. Is it devaluing the natural human reproductive process? We must ask, if the model develops organs and function, at what point does it acquire moral status? Where is the dividing line between treating these lab-grown, lifelike entities and treating mere cell clusters? The challenge to the 14-day rule and critical thinking. Globally, the unwritten rule of the 14-day rule has been an ethical red line, restricting the culture of human embryos to prevent study after the nervous system begins to form. Yet Hanna's model circumvents this rule entirely, as it is derived from stem cells. From a critical thinking perspective, this suggests that the rulemakers may have been outpaced by science. Some question, are we deliberately obstructing research that could save millions of lives simply because we fear the ethical ramifications? Or will the collapse of the 14-day rule truly open the floodgates for unimaginable forms of biological engineering? Scrutinizing scientists' self-regulation, Professor Hanna and his team emphasize that their goal is to understand, not create, life, and they claim strict adherence to existing guidelines. but we must scrutinize this. How long can scientists' self-regulation truly last? When immense commercial value, medical urgency, or even military potential emerges, will reliance on moral self-restraint be sufficient? Should we start now to establish an independent, transnational ethical oversight body to govern this powerful technology that has the potential to redefine humanity itself? This is a question demanding a serious answer from all of us. V. Conclusion and call to action. The Mystery of Life, summarizing the significance of the research. Jacob Hanna's work is undoubtedly a milestone in human self-understanding. It is no longer just observing nature, but beginning to deconstruct and reshape the most basic blueprint of life. It opens a door for us to see every detail, every error, in embryonic development. Final thoughts. As the scientific community continually pushes limits, these synthetic models will become increasingly complex. This forces us to ponder an ultimate question. What exactly is the essence of life? Where are the boundaries of technology? Call to action. This is an issue that requires attention from all of us. Do you believe Hanna's research is humanity's hope or a potential Pandora's box? Leave your opinion in the comments section and join this grand debate about the future. Don't forget to like and subscribe. We will continue to track the forefront of life sciences for you.